Alternation of generations is kind of a big deal in plant biology. For one thing, it gives us something to keep track of as we survey the plant kingdom, from the simplest mosses to the most derived orchid. In order to grasp the specialness of alternation of generations, we should start by thinking about diploid and haploid parts of the life cycle in a sexual animal. A human will work well enough as an example here. The diploid multicellular adult produces haploid unicellular gametes, sperm or eggs, and during fertilization, these fuse to make a unicellular diploid zygote. Zygote grows into the diploid multicellular adult, and this completes the cycle. Note that the haploid part of the cycle is exclusively unicellular. Now in plants, there are both diploid and haploid generations involving unicells growing into adult multicellular plants. If we start with two gametes, still sperm and egg, fusing to form a diploid zygote, this zygote grows into a multicellular plant that's diploid, the sporophyte. Somewhere on the sporophyte is a specialized organ, the sporangium, where cells undergo meiosis to produce haploid cells. And these are spores, not gametes. Remember, a sporophyte, the plant, will bear sporangia, the organs, which bear spores, the haploid unicells. Unlike gametes, spores do not fuse with other cells, and they remain haploid and grow by mitotic cell divisions into a multicellular haploid plant, the gametophyte. Somewhere on the gametophyte is a specialized organ, the gametangium, that bears haploid gametes. Remember, gametophyte, the plant, will bear gametangia, the organs, which bear gametes, the haploid unicells. Now, the gametophyte is already haploid, so when it produces haploid gametes, it does so with a mitotic division. Meiosis happened back when the diploid sporophyte produced the spores. Plants, like animals, are oogamous, meaning they have differentiated gametes that are either eggs or sperm. This requires that the gametangia producing the two kinds of gametes are different. Egg gametes are produced at egg-specialized gametangia called archegonia. Sperm gametes are produced at sperm-specialized gametangia called antheridia. Once the gametes are formed, the cycle can then complete with sperm and egg fusing to form a diploid zygote. This is an alternation of generations in the sense that you have two generations of multicellular plant, the sporophyte and gametophyte, that alternate in sequence. The sporophyte has a child that grows from the unicellular haploid spore, and that child is a multicellular haploid gametophyte. The gametophyte successfully reproduces when one of its gametes fuse with another gamete to make a zygote, which then grows into the gametophyte's multicellular child, which is a diploid sporophyte. In land plants, the sporophyte and gametophyte always develop differently and are distinguishable. In lab, we identify and document in our lab notebooks the sporophytes and gametophytes for mosses and ferns, and this should give us a clear picture of how alternation of generations works. Then later, as we look at the more highly derived situations seen in seed plants, we can still use our understanding of the moss and fern gametophytes as a conceptual anchor as we follow the trail of crumbs to what's left of the alternation of generations in seed plants. First gymnosperms and then flowering plants. After mosses and ferns, things tend to get a lot more challenging, and it's important to have the reference point of these lessons from simpler plants. As I said before, you'll need to distinguish between two types of gametangia on the gametophytes: archegonia for the production of eggs and antheridia for the production of sperm. By the way, angium means vessel, as in container. So gametangia are the containers for gametes, just as sporangia are the containers for spores. Telling the two types of gametangia apart is actually pretty easy on mosses and ferns. You'll see that each antheridium contains multiple sperm, thousands in the case of mosses and dozens in the case of ferns, while an archegonium will contain just a single egg surrounded by nurturing support tissue. The reason for this is tied to the concept of land plants being embryophytes. Of the two kinds of gametes, only the sperm is released from the gametophyte. The egg remains in the archegonium, that is, attached to the mother gametophyte, and fertilization takes place when the sperm arrives at the doorstep of the egg. at the archegonium of the gametophyte that will become the mother or the egg parent of the resulting progeny. After the egg is fertilized by the sperm and the resulting zygote begins to grow into a sporophyte, it remains attached to the mother gametophyte, drawing nutrients from it. This phenomenon of having the sporophytic embryo nurtured by the mother gametophyte is a synapomorphy of the plant kingdom. The other name for the kingdom Plantae is the Embryophyta, named for the fact that sporophytic plants develop as embryos nurtured by the gametophytic parent. This happens in a very obvious way in mosses, and you should be able to visualize this embryophyte situation based on what we saw in the laboratory. Sporophytes still being attached to gametophytes at the location where the nurturing tissue of the archegonium interfaced with the zygote. In ferns, the gametophyte, or prothallus, is tiny, and we only saw them in the prepared slides. In theory, you could go out into nature and collect sporophyte and gametophyte as attached entities, but you'd have to be lucky enough to catch them just at the stage where the sporophyte is starting to grow. Shortly after this, the gametophyte gets entirely consumed by its baby, the little fern plant, which then thanks its mother for the good head start it got in life from the resources provided by its gametophyte mother's body. Yes, mom is here the baby's first meal, and once mom gets completely consumed, the young fern becomes independent, making the raw materials for its growth via photosynthesis. In an earlier lecture, we've talked about the features of plants that made them successful on dry land. Sporopollenin, for example, sealed off plant tissue, keeping it from dehydrating. Vascular tissue was the other obviously successful innovation, as it allowed plants to access soil water and to grow to large size. Neither of these requires further explanation. The embryophyte condition was also instrumental in allowing plants to invade a harsh new world where drying out and limited access to favorable habitats were challenges that had never been seen by the plant's exclusively aquatic predecessors. Remember that spores grow into gametophytes, and then for the next generation, gametes make zygotes that grow into sporophytes. To make one complete turn of the alternation of generations requires 2 periods of growth from almost nothing. Unicellular spores or zygotes have just a tiny amount of resources, and they have to fuel most of the growth from photosynthesis. But being tiny means that you have very little photosynthetic tissue, and so the rate of growth is going to be extremely slow until the plant gets bigger. Now think about what embryophytes do. Their zygotes make use of the resources already there in the gametophytic mother, and this means that the sporophyte can grow much faster. Early land plants still had to grow their gametophyte from spore, but being an embryophyte meant shortcutting what would otherwise be a slow initial growth phase of an independent zygote. The contrast here can be seen with green algae, which have alternation generations, but they release all their gametes into water, eggs and sperm. In other words, they're not doing the embryophyte thing where the archegonia retain the eggs. In the algae, fertilization occurs in the water column, and zygotes grow from tiny unicells into adult sporophytes. This is a stage that land plants shortcut by being embryophytes. But it works well enough for the green algae because its aquatic world is benign, that is, relatively conducive to the growth of tiny things, and by producing enough of the eggs, sometimes numbering in the millions. Fertilization would thereby result in millions of zygotes, and there's a reasonably good chance that at least some of them would be successful in reaching adulthood. Living full-time as aquatic algae means that you don't have to worry about the availability of moisture that may seasonally vary on dry land. For plants, however, there is the constraint of length of time that moist conditions are available for plant growth. If the spore to gametophyte growth phase begins with the onset of the wet season, there might not be enough time remaining after that to grow a full sporophyte from a tiny zygote. The embryophyte solution to this problem is basically to take the nutritional resources there in the body of the gametophytic mother, acquired by photosynthesis over her growth from a spore, and recycling these resources by pumping them into the baby sporophyte, which then gets to a larger size more quickly thanks to this head start. I want you to think about this. Remember that it's an anchor for our later discussions on the more highly derived seed plants. How does this characteristic of being an embryophyte relate to the invasion of dry land by plants? Being able to explain this part of the narrative will require you to integrate concepts of the comparative ecologies of aquatic versus dryland habitats with the alternation generations we learned about in the first part of this lecture.